And so we would come back to finish off with Professor Hanu Pappu's presentation. Professor Pappu is the President Sam Smith Distinguished Professor and the Chuey Endowed Chair in the Department of Plant Pathology. And he is also a member of the interdisciplinary PhD program in molecular plant sciences. And he will be talking about harnessing AI for food security.
Right. Good evening, everybody. Thanks for sticking around, everybody. So first few minutes, I'll briefly talk about the global disease burden and how AI can really address this issue. And a couple of ongoing disease outbreaks, we call them COVIDs of crops. And then give a broader perspective about use of AI in agriculture and crop production. Manoj gave a very nice overview of AI in crop production practices. And so I will specifically talk about a case study of AI in plant pathology in my own research program, how we are studying specifically about viruses that cause serious diseases in many crops, and how we're trying to use AI in understanding how viruses interact with their plant hosts and cause disease and cause damage. Okay? Usually I run out of time, so I would like to first thank my team, a team of 3 postdoctoral fellows, Hira Kamal, Ingzai, and Prabhu, very capable and productive postdocs, and my collaborators, Imran Amin, who is a plant virologist, and Dr. Minhas, he's a bioinformatics and AI person. And most of the work that I'm going to talk about is done by my postdocs, and I really like them because they do all the work and they let me take the credit. So, and most of my research I'm going to show is supported by USDA, NSF, and WSDA, and a Chuey Charitable Foundation. So I thank them for the dollars they gave me. So what is food security? Global population is projected to reach 9 billion by 2050. And that is compounded by the fact that increased urbanization, loss of agricultural land, and water becoming limited. It's going to be a huge challenge. And right now, 1 in 5 can't afford to have 3 meals, and millions go hungry every day. And malnutrition is a major impediment for quality of life. And United Nations and FAO studies have shown again and again, civil strife that we see is directly linked to poverty and insufficient food and water. Here in Washington and in the country, a lot of important crops are affected by many diseases. Just here in Washington, the climate is very conducive to disease outbreaks. And Washington has a very diverse agriculture. We grow literally from A to Z, or in this case, A to W, apples to wheat, very diverse agriculture, a multi-billion-dollar industry. And there are hundreds of diseases that affect both quality and the yield of these crops. And of course, the last one in the list in the crops, hops. I think as we are getting closer to the evening, I'm showing more interest in hops, one of my favorite crops, ready for a glass of beer. Speaking about staple crops that sustain life, there's only about a dozen of them that sustain life, in fact, on this planet. And so I'm going to quickly use a couple of examples of how these staples are affected by diseases and how AI can really make a positive contribution. One is potato in a historical context, and then I'll talk about an ongoing disease outbreak in corn and in one plantain. Most of you probably have seen this movie Martian. Our hero gets stranded on Mars, but manages to survive because, spoiler alert, he had some potatoes left with him, and that sustains him. So he figures out how to grow them because he's a botanist by training, luckily. But potato has a very long history, and the disease of a potato actually affected the human civilization, migration, and mass movement of millions of people from Ireland to US in 1800s. And most of you are familiar with the Irish potato famine. Potato is a major staple, major food crop, and a disease outbreak over a period of 5 years, year after year, destroyed the crop, causing millions of deaths. And here is a rotten potatoes, both in the field and in the storage. Millions of deaths and millions of migration. And this particular late blight of potato still continues to be a constraint to potato production in many parts of the world, even after all these years. But it is being managed very well with fungicides. In the foreground, untreated; in the background, it's treated by a fungicide. But the problem is that this late blight pathogen is developing resistance to these chemicals, meaning these fungicides are losing efficacy. So here, in fact, many chemical companies are using AI and ML to screen compounds whose structures are known, hundreds and thousands of compounds, and shortlist them or narrow them down to those that can have potential fungicidal activity so that they can actually be tested. So from thousands to a handful, for example. Similar to some of the pharmaceutical companies using AI to screen thousands of compounds and select few that have pharmaceutical value potentially. Right? So this is where AI is now being used as I speak to come up with chemicals that with a potential to control these diseases. Bananas, most of us eat almost every day. Cavendish is the banana variety that pretty much people eat all over the world, and it's a clone. So every banana is identical to another banana. It's identical. So as a result, the downside is that if one banana plant is affected by or susceptible to a fungus or a pathogen, pretty much every banana plant is expected to be susceptible. So an outbreak of a disease called Panama disease can destroy potentially the bananas as we know. And a new strain showed up called Tropical Rest 4. And now it is spreading all over the world. Most recently in the Western Hemisphere, it was found in Colombia. So there are no control options. Many labs are now using many different approaches, including genome editing and incorporating AI and ML in trying to identify what genes could potentially be targeted in this fungal pathogen and also in banana and edit those genomes and see if we can come up with a control option. The race is on, and without control options, a dire prediction is that banana could become a rare commodity. In Africa, corn is a staple crop. It's a very important part of the diet, especially in Eastern Africa. And Kenya is a good example. And few years ago, a new viral disease broke, epidemic virtually destroyed millions of hectares of corn. And it continues to be a major problem to corn production in East Africa. First reported in Kenya, and now it spread to several East African countries and affecting livelihoods, because in Africa, it's a smallholder agriculture, subsistence farming. Farmers grow and they eat what they grow and then sell what is left. So it is a livelihood that is being affected by this disease. And again, I am involved in an international project where we are now looking at both corn genome and the viral genome and using AI to predict what are the proteins that are interacting, resulting in this very destructive disease. So coming to AI in agriculture, it has tremendous potential. Manoj alluded to some. Robotic applications are numerous in harvesting crops, and deep learning algorithms for drones. There's quite a bit of research is going on in that area at WSU, and machine learning models to predict environmental impact on crop yields, environmental impact like climate change. So AI has a tremendous potential in all these areas of agricultural production. And smart farming, we call it, using AI and ML in crop management, predicting yield in a given region, a given farm, and predicting the crop quality and detecting diseases and weeds. And again, Manoj commented about that. And various fields in agriculture crop production, pest management, genetics, horticultural production, and plant breeding. AI has tremendous potential and is being used for agriculture enhancement. And at field level, again, in addition to what Manoj talked about, managing soil, managing water, also there's quite a few reports coming out using AI. Specifically in plant pathology, that is managing diseases. There's quite a bit of progress has been made using AI in terms of disease diagnosis. Now, several applications are freely available that make use of AI in disease diagnosis and incorporated into handheld devices like smartphones, where you take a picture of a diseased plant in the field in the middle of nowhere, and you upload it to a server, and then you get almost instantly a diagnosis. So that is now, it became very handy and commonplace in many countries because the penetration of smartphone technology is very high, whether it's in most parts of the world. So this is a busy slide, but basically applications of AI and ML in plant pathology is happening at the field level. And both at the field level and also at the molecular or fundamental level of research, understanding how pathogens and cause disease and what is the mechanism behind it. And AI has made a significant contributions, and I'm going to give one example. So in my lab, specifically, we've been studying virus-host interactions at the molecular level, looking at the transcriptomics, that is what kind of transcriptome changes are happening in the host plant as a result of a virus infection, and also the protein profiling and metabolomic profiling, what are the changes happening in the host. And we are using genome editing for trait improvement. So I'm going to use specifically one disease as an example. That's a project that's ongoing in my lab is on cotton, is a multi-billion-dollar enterprise. And there's a very serious virus affecting cotton production worldwide, is cotton leaf curl. Interestingly, cotton leaf curl, this virus is a DNA virus as opposed to COVID, which is an RNA virus. So this DNA virus codes for a particular protein that we know that it causes the disease symptoms. And so we want to know how this viral protein interacts with the cotton plant when the virus enters the cotton plant. And cotton, typically like most plants, they code for thousands of proteins. So it is basically looking for a needle in the haystack, which protein or proteins in the plant are being affected by this viral protein. So initially we used AI to screen these thousands of cotton proteins and narrowed down a few that could be potential targets for this viral protein. And it turned out that one of the cotton proteins is called kinase, is a potential target for this viral protein. So the hypothesis is that this viral protein is selectively targeting binding to this cotton protein, so disturbing the plant metabolism, the plant growth, and the physiology, and leading to disease and yield loss. So that was the hypothesis. So, but we wanted to know what parts of this cotton viral protein, in fact, is selectively targeting what domains of the plant protein. The idea is that once we identify those, we can come up with targeted therapies by designer molecules that would block this interaction between viral protein and the plant protein. Meaning we are trying to then suppress the virus replication, less disease, less impact. So that is the goal. So we used a suite of software that make use of AI to identify these interacting partners between a viral protein and a plant protein. Because we are, again, looking at thousands of proteins in the plant. And we used, again, a suite of programs, first for sequence recognition, that is the viral protein sequence and the plant protein sequence, do they recognize each other? And then what are the binding sites in these two proteins that actually come together and bind to each other? and what are the specific amino acid residues within this each protein that in fact physically interact, and that interaction resulting in the viral protein interrupting the activity of the plant protein, affecting the plant metabolism. Right? So all these programs essentially gave us some ideas, and my postdoc insisted that I show this slide. It's a very busy slide, but this number of different software, the programs that we used to identify, to narrow down potential interacting sites. So we are looking at a single amino acid in a protein consisting of thousands of amino acids. So it's a one bead in a necklace, sort of. So it turned out that top left is a cotton plant protein, and the bottom right is the viral protein. And using this software, we identified that certain domains of this protein are likely to interact with the plant protein. And further analysis turned out that 2 of the 4, UBA and AIS, are more likely to physically bind to the cotton protein, making it inactive, leading to disease ultimately. So, and then the question is, what specific amino acids within these domains are critical? Again, the idea is that once we identify those amino acids that are critical for the activity of the viral protein, we can come up with, use a genome editing technology to modify that. Or we can come up with a designer RNA molecules to silence that gene, what you call gene silencing. So the applications are numerous once we have this information. So then another program software we used actually were able to predict that certain amino acids in this viral protein have propensity or affinity to bind to the plant protein. And the next step would be experimental validation. This is all prediction, but experimentally then my postdoc went ahead and actually physically showed by expressing these viral protein and the plant protein in a plant cell. And when these proteins physically interact, they fluoresce because each protein is tagged with a fluorescent marker. So every time they physically interact, you see fluorescence. So she actually experimentally validated what these software predicted as a potential interacting partners. So she used a different set of experiments to show that physically, yes, in this case, if the proteins interact inside a yeast cell, thus yeast can grow. If they don't interact, the yeast cells won't grow. So a very simple scoreable system. So basically, this is just one example of how AI can really give us clues in predicting potential partners in plant pathogen interactions, so that then we can experimentally validate. And now the next step that we are working on is to come up with these designer targeted therapies, if you will, to intervene in this viral and plant protein binding sites, so that the virus replication is suppressed, resulting in resistance to that virus. So future of AI, linking some of the sensor signals to processes during plant pathogen interactions has a huge potential, and development of a scale and environmentally independent features, integrating sensors such as 3D imaging sensors to assess the complex architecture. So the applications of AI in agriculture and specifically in disease management, they are numerous, and this is only the beginning. So I'll stop there and answer any questions you may have.
Thank you very much, Hanu, for this wonderful presentation. It was worth waiting, and I'm sure many of the people who are attending would agree with me. I have several things that I would like to follow up on. We are past 5, about 15 minutes over, and thank you for sticking around. So let me thank all the speakers for the time and for your sharing great presentations. This was a conversation starter for many of us, and I hope this will be used for that purpose. Allowing the presenters, the slides will be available on the website, and the recording will also be made available. And for those who have attended, we will follow up with anything that would happen around this one. This is Research Week. Tomorrow there are more events happening, including the Distinguished Vice Presidential Lecture. And I would like to encourage you to go there, find information and attend. And once again, thank you very much for sticking around this long. And I appreciate your attendance. And I deeply, deeply appreciate those who spoke for your time and the things that you've shared. I will see you around. Thank you.